Hello students, in this video we'll discuss about the meiosis. Meiosis first. In this there are phases, as we know already. In previous video we have discussed the phases of meiosis first, that is prophase first. In this prophase, again 5 sub phases are there. First one is the leptotin, so that is called the leptotin phase. Leptotin or leptonema. Next one, zygotin, and third is pakitin. Fourth is diplotin and diokinesis. Diplotin, and final, that is last phase or fifth subphase of the prophase first is diokinesis. These are the 5 phases. So in previous video we come across leptotin, zygotin and even pakitin. Once again we see these phases after we're going to discuss about diplotin and diokinesis. So the first one I'll choose here, the leptotin only. Leptotin. Yes. In this leptotin, the actually chromosomes starts appearing because of condensation of chromatin material. Condensation of chromatin to form chromosomes. Chromosomes are formed and they are become visible in the microscope. So but chromatids are not clear yet. So here when you observe the diagram of this one, this is the nucleus, and this I will consider it as the cell, right? So in this nucleus there is chromosomes are elongated and thread like, right? So like this can show the chromosomes, right? So these are the chromosomes I have chosen here. They are elongated, right? And this stage shows that the chromosomes are elongated and Bouquet stage. So we may also refer it as Bouquet stage. Condensation of, the first point is that condensation of chromatin leads to formation of the chromosomes, and there is a so you can observe here clearly the condensation of chromosomes. And next, the centrioles develops esters. Centrioles develops esters, right? So that means here you can show the esters, and it also divides and move, that centrioles are moved towards the opposite poles. This is what that moving towards the opposite pole, right? So this is what the leptotin. And next I'll choose, even this leptotin is also called a Bouquet stage. You can remember Bouquet stage because these chromosomes are so appear like this. They are attached to the nuclear membrane, and actually in animal cells chromosomes are converged towards the centrosomal ends to form this Bouquet stage, right? And the ends of chromosomes are attached to the nuclear envelope by a structure called attachment plate. So this is what the somewhat attachment plate like structure form. That's what we call it as a bucket stage, right? So next we go to the next subphase, that is zygote. So in this zygote, the homologous chromosomes undergo pairing. The first point, homologous chromosomes undergo pairing. Homologous chromosomes undergo pairing. Here, this process is called synapsis. The synapsis takes place here. So that means homologous chromosomes means the morphologically similar chromosomes are called homologous chromosomes. This is what the cell, this is the nucleus, and you can show the esters here. They are starts moving here. Yes, these are the, you can also mention it as a ester chromosomes, and this is the nuclear membrane. Here also you can mention the same thing. This is the cell membrane, nuclear membrane, ester, cell membrane, that is nuclear membrane, and ester, and this is what the homologous chromosomes here you can observe. Right, homologous chromosomes. Right, so these are referred as the homologous chromosomes, right? So this pairing of homologous chromosomes is called synapsis, right? So here in zygote we observe that. In the next stage, even the homologous chromosomes are actually hold together to maintain their alignment by synaptonymal complex. What do you call? Synaptonymal complex. Synaptonymal complex is formed here to maintain this alignment between the two homologous chromosomes, right? In between these two chromosomes, there is synaptonymal complex is formed by a specialized protein which will held together these two chromosomes, right? And these homologous chromosomes are called bivalents. Now we call it as a bivalents, right? So actually these are, when we observe very clearly in the next stage we can observe. So this chromatid splits to form two arms. So here, so you can mention the two arms here, and for understanding you can draw it in the passaging stage. See, I'll go to the next passaging or packeting. Here paired chromosomes become thick. Here paired chromosomes become thick and short. What we observed here, elongated, no, in the first phase leptotin, they are still condensation, and finally in the passaging, the highly thick and short they are become. So I'll show, this is the nucleus now, and this is the cell, right? So here the chromosomes are, they appears like this, right? And even we can show the crossing over here. These are the homologous chromosomes here. and one more pair you can show here. Yes, so by this we can understand the crossing over. This is a X-shaped structure is formed. This is called chiasmata. And this process is called crossing over. The exchange of genetic material between two homologous chromosomes, that is between usually non-sister chromatids, right? So that process is called crossing over, right? So the exchange of genetic material between two homologous chromosomes is called crossing over, right? The crossing over is very important process here in the Patcherton stage, that may be asked for 1 marks also. So you can remember, exchange of genetic material between two homologous chromosomes is called crossing over. Exchange of genetic material between two homologous chromosomes is known as crossing over, right? Two homologous chromosomes, it is two homologous chromosomes. In between two homologous chromosomes, here you can observe, due to X-shaped structure, that is with the help of a protein called endonuclease, and one more is ligase. So both endonuclease helps to cut this the arms of this chromatid, and ligase helps to join this. So by this, what happens here, the two exchange of chromatid parts takes place in this point. That point is called chiasmata, right? So X-shaped structure is formed now, so that is called chiasmata, right? So here in the Patcherton stage, very important thing is paired chromosomes are called homologous chromosomes, and they are become thick and short, right? So here in this stage only the exchange takes place. The exchange of genetic material takes place between the two this homologous chromosomes, and now we call it as bivalent also. So this is what we referred. So this is the bivalent, right? So the bivalent means here the paired homologous chromosomes are called bivalents. Paired homologous chromosomes are called bivalents, and the each bivalent means this paired homologous chromosomes has a tetrad. So what we call tetrad, there are 4 chromatids of bivalents are called that is bivalents are called tetrad, right? So the tetrad is formed here in this Patcherton stage, and in the diplotin stage. So next we go for the diplotin. So I'll take here in the next page, that is in diplotin stage, the synaptonymal complex is dissolved synaptonymal complex is dissolved, and the homologous chromosomes repel each other. Homologous chromosomes repel each other, so that is the repulsion. That is called repulsion. They start separating from each other. That is known as repulsion. It takes place, to which the homologous chromosomes are attached at certain X-shaped regions. No, that is called chiasmata. So that still appears here, chiasmata. Chiasmata we call, right? So here the structure, when you observe, this is the cell, and the nuclear membrane starts disappear, and nucleolus is also disappeared. So only S stars you can show here. S stars you can show, right? And these are the chromosomes you can show here, like this. There are stars appearing in the chiasmata region, right? Chiasmata, the X-shaped structure, no? This is called the chiasmata. This is one chromosome of bivalent chromosome, right? So this is the exchange X-shaped structure what we are observing here. This is called the chiasmata, right? So next one more pair of bivalent I am showing here. Right, here also X-shaped structure, that is called chiasmata, right? So here this is called the chiasmata, and these are stars repelling each other. Okay, so and in diplotin stage, usually in female side, so what happens, which is arrested for 8 to 10 years, that what we observed, right? So from fetal stage to 12th year, during up to ovulation, it may be arrested. So this is, you can mention it as chiasmata, right? So next stage is, so I'll go for the 5th stage, that is the Diokinesis. So in Diokinesis, so we can observe that the highest, the chromosomes are thickened, right? Chromosomes are become very thick and short. So in this stage, chromosomes become highly thick and short, right? So next, so when you observe the diagram here, so this repulsion is continuous. The homologous chromosomes are still repelling each other, and the exchange of genetic material takes place. So we can take this diagram here. So now we can show this structure, that X-shaped structures are repel each other. This is the chromosome exchange part, right? So here, from this chromatid part, this is passed here, the exchange of chromosome. So you can observe here very clearly the diagram, right? In the same way here also. This portion can show it. This is the exchange of genetic material, is very clear, it takes place here. So this is what the actually homologous chromosomes, they repel each other, and this is what the astral rays are formed here. Astral rays, we can say. Astral rays are formed, And in this terminalization is the process which is occurs. What is the meaning of terminalization? The chiasmata slide towards the tip and disappears. Chiasmata are disappeared here, or disappeared. Chiasmata are disappeared here. So the chiasmata when disappeared, no, that X-shaped structure is disappeared. That is called the repulsion is to go on continuous, and bivalents are repelling each other, right? So fully condensed, and they are moving towards the tip of chromatids, and terminalization is takes place. Terminalization, and process is called terminalization. The terminalization is also takes place here. So means the X-shaped structure gets separated, exchange of genetic materials takes place. This is all with the help of the ligase and endonuclease. That is what the enzymes which are involved in it, ligase and endonuclease. And nuclear membrane and nucleolus are disappears here. Very important. Nuclear membrane and nucleolus both are disappeared continuously, completely, sorry. And spindle fibers are begins to form. Spindle fibers are formed. They are starts producing the spindle fibers like this, right? So this is what the Diokinesis, and then it enters the next phase, that is called we refer it as this is the prophase. Next one is metaphase. So prophase first is over now. In the metaphase we will discuss in the next video. Thank you, students.